There's something your body does in the dark that it never announces. No sensation accompanies it. No shift in temperature. No sound. No signal you could name if you tried. But something begins. Something that was never not happening, really. And the quiet of wherever you are right now is already part of it. Welcome to the Sleepy Biologist. If you've been here before quietly, thank you. If tonight is your first time settling in with this channel, then you arrived on a good night. The people who've made memberships and left kind words and pressed the like button without being asked for it. They're part of why this exists, this space and the time it takes to build it. I don't take that lightly, now, before eyes, before the nerve cell that would eventually learn to carry a signal from one tissue to another. Before anything you'd recognize as sensing, there was already a surface, and that surface was already doing something with light. This is the part that tends to get skipped over, even in careful biology. We talk about eyes as though vision were the beginning of the story, but eyes are almost embarrassingly recent, in the timescale of things. What came before them was something slower and stranger. A protein called opsin, embedded in cell membranes, capable of changing shape when struck by a photon. No image, no signal forwarded anywhere meaningful, just change. A molecular response to the presence or absence of light, folded into the membrane of a cell that had no particular name, in a body that had no particular plan. Opsins like this still exist in your skin. That sentence is easy to read past. Read it again, maybe. Photoreceptive proteins in skin, in the gut lining, in certain immune cells. Not functioning as vision, there's no pathway here that sends light information to any visual processing center. But responding, registering, doing something quiet and chemical with the information that photons are present, or that they are not. What they do with that information is still being worked out. There's something honest about admitting that. A lot of the biology in this chapter lives at the edge of what's been confirmed and what's suspected. The opsins in skin, particularly OPN3 neurosin, have been studied in relation to things like UVB radiation responses, pigmentation, peripheral circadian timekeeping. Some evidence suggests they contribute to a kind of local clock, a tissue-level timekeeping that doesn't wait for instructions from the brain. The skin keeping its own quiet time. It's not entirely clear how much this matters compared to the retinal signals that drive your central circadian rhythm. It may matter very little, but the existence of these proteins, the fact that they're there, old and patient, doing something, suggests that your relationship with light and darkness is layered in a way you don't normally feel. There's the version you experience, dusk, the lamp switching off, the room going dark. And then there's this other version, older and slower, that your surface is engaged in without asking you. 300 million years is hard to feel. Maybe more than that, depending on which lineage you're tracing, which organism you're using as your reference point. There are estimates that opsin-based photoreception predates complex eyes by something like that margin, which means the skin-level response to light isn't a footnote to the story of vision, it's an earlier chapter of it. The eye is the specialization. The membrane protein is the original condition. Right now, in the dark or the near dark of wherever you've settled, your skin is not just a boundary. It's something that has been registering the state of the world for longer than you have language to describe it. The cooling that happens at night, the shift in ambient light, the particular quality of stillness that comes after a certain hour. Some part of your surface has been tracking this, not as experience, as chemistry, as the slow accumulation of molecular states that say, this is what night feels like, this far from the sun, at this hour, in the body. You probably noticed the weight of the blanket before you noticed anything else tonight. the specific press of it. That sensation has nothing to do with any of this, and yet it arrived at exactly the right time, the body already doing what bodies do in the dark, already leaning towards something it doesn't have to decide to do. Darkness isn't what happens when light leaves. It's a condition the body has been preparing for across every hour of the day, every gradual shift in photon count, every tick of a protein clock in the epithelial layer, every upstream adjustment in a cascade that won't complete for hours yet. The skin knew this was coming. It's known for a long time. Something in the brain's older corridors is already receiving that message, already beginning the translation from surface chemistry into the next deeper sequence, not of sensation, but of signal, the kind that travels inward. The lights go out and something happens before you decide anything. That's the part worth sitting with. Not the fear exactly, that fear might be too large a word for it, more like a readiness, a shift in the quality of your attention. The room is the same room. Nothing has entered it. And yet something in the processing has changed in the time it took for the light to leave. You didn't choose that. It arrived ahead of you. There is a structure deep in the temporal lobe, roughly almond-shaped, present in both hemispheres, that processes threat-relevant information faster than the cortex can form an opinion about it. The amygdala. You've probably heard its name before, usually in the context of stress or anxiety, usually in a way that makes it sound like a problem. It isn't a problem. It's old machinery doing what old machinery does, running the same program it's been running for a very long time, in a very different set of environments than the one you're currently lying in. What it receives from the retina is not an image. That comes later, after the visual cortex has done its processing, after shapes and distances have been resolved into something recognizable. What the amygdala receives, through a faster, rougher pathway, sometimes called the low road, is coarser than that. Brightness levels, contrast ratios, the sudden drop in photon input that follows when a light source is removed. Enough, apparently. Enough to act on. The response is not dramatic. That's what tends to get lost in explanations of this. There's no alarm sounding, no single moment of fear that announces itself cleanly. What happens is more like a change in threshold. The amygdala lowers the bar for what it will flag as worth attending to. Sounds that were unremarkable a moment ago are now slightly more salient. Movement at the periphery, the creak of a house settling, none of that changed. Your evaluation of it did. And beneath the evaluation, briefer than you could track, there was a moment, probably somewhere between 80 and 120 milliseconds after the light dropped, when the amygdala had already begun forwarding a signal. To the hypothalamus, to brainstem nuclei involved in arousal, to the systems that coordinate the body's readiness posture. The cortex was still constructing the thought. It got dark when the subcortical response was already underway. This is not anxiety, it's latency, the gap between what the body does and what the mind catches up to. It's worth saying something about where this comes from. Not the amygdala specifically, that's a mammalian structure, relatively recent, but the reflex underneath it. The shift in alertness that darkness triggers is documented across vertebrates, birds, fish, reptiles whose nervous systems look almost nothing like yours. Something about the withdrawal of light changes behavioral state broadly across a wide range of organisms. The amygdala is your version of a much older mechanism. It inherited the job from circuits that predated it by a significant margin. Predators in most ecosystems are more active after dark, or were. Across the long stretch of time when the behavior being encoded it was actually being selected for. In those environments, a nervous system that relaxed when darkness arrived was not, on average, a nervous system that left descendants. The ones that persisted were the ones that treated dark as a condition requiring slightly more vigilance. That's all this is, a record of an old environment running in a body that no longer lives there. The strange part, and this is the part I find myself returning to, is that the record runs whether or not it's relevant. The amygdala doesn't check whether you are in a modern room with locked doors and no predators. It doesn't have access to that context, not at the speed it operates. By the time the prefrontal cortex has assembled a calm, rational assessment of the situation, the subcortical response has already moved through several stages. You can feel the cortex arriving late sometimes. That slight unnecessary alertness when the room goes dark. The way it dissolves, usually within a few minutes, as the higher systems catch up and override. That dissolution is real. The prefrontal cortex does actively dampen amygdala activity through inhibitory projections, through the gradual accumulation of evidence that nothing is wrong. But the first move was always the amygdala's. Right now, if there was a flicker of that earlier, a small tightening when you switch the light off, a brief inventory of the room's sounds, that was this. That was a structure in your temporal lobe doing something it has been doing in one form or another for longer than your species has existed. It's not a flaw in the system, it's the system, and it quiets. The inputs stop arriving with the same urgency. The arousal signal loses momentum without new evidence to sustain it. The cortex sends its slow reassurance downward through inhibitory pathways. The threshold climbs back up. Sounds return to unremarkable. This is already happening. The readiness is already draining out. Somewhere upstream of all of it, in the retina, in the cells that track not images but duration. Not what the eye sees, but how long the dark has been present. A different kind of signal is accumulating, one that moves on a different timescale entirely, not milliseconds, hours. Most of what your eye does happens quickly. Shapes resolving, distances collapsing into perception, movement tracked and categorized before you finish the thought that you saw something. That part of vision is fast, impatient almost, always updating. But there is another thing the eye does that has nothing to do with any of that, and it is very, very slow. Buried in the retina, not in the rods, not in the cones, but in a third class of cell identified less than 30 years ago, there are neurons that don't build images. They are called intrinsically photosensitive retinal ganglion cells, which is a long name for something that does a remarkably quiet job. They contain a photopigment called melanopsin, and melanopsin, unlike the pigments in your rods and cones, is not optimized for rapid response. It doesn't need to be. It isn't tracking what's in front of you. It's tracking something much slower. It's tracking whether it's day or night. These cells, sometimes written as iPRGCs, if you've come across that abbreviation anywhere, respond to sustained light over long durations, not flashes, not movement, not contrast or detail. They integrate photon input across minutes, sometimes longer, building a slow chemical average of how much light has been present. And for how long? A single brief bright flash barely registers to them. But 2 hours of moderate indoor lighting? That accumulates. That gets counted. Their axons don't run to the visual cortex. They don't contribute to what you see in any ordinary sense. They run instead to a small paired structure in the hypothalamus called the suprachiasmatic nucleus. About 20,000 neurons in total, sitting just above the point where the optic nerves cross, and serving as the brain's primary circadian pacemaker. The iPRGCs report to this structure. They tell it, in the only language they have, how bright it has been and for how long. The suprachiasmatic nucleus listens and adjusts. There is something perfectly remarkable about the scale mismatch here, though I want to say it carefully without making it bigger than it is. 20,000 neurons. in a brain of roughly 86 billion, the circadian pacemaker, the structure that coordinates the timing of almost every major physiological process in your body, that synchronizes hormone release and core temperature and immune function and cell division across dozens of tissues, runs on 20,000 neurons. Less than a street block of cells doing that. The iPRGCs feeding into it are fewer still. There are roughly 3,000 of them in the human retina. 3,000 cells integrating your light exposure over hours, forwarding a signal to a tiny nucleus, which then signals outward to the rest of the body through a combination of neural and hormonal pathways. The pineal gland receives the message. Your liver receives a version of it. Your skin, in a looser downstream way, receives an echo of it. All from 3,000 cells in the back of each eye, doing something most people have never heard of. Right now, if the light around you has been low for a while, if the lamps are off, if the screen you might be listening from has been dimmed, if the natural dark has been present for more than half an hour, those cells have been integrating that absence. They don't alert you to it. They don't produce a sensation. There's no feeling of the slow clock ticking, but the signal is moving. The suprachiasmatic nucleus has been receiving across the past several hours a gradually shifting report. Less light than there was, then less than that. The pacemaker interprets this as the approach of biological night, not the night outside exactly, but the body's internal version of it, which can be moved and manipulated by light in ways that feel almost disconcerting once you understand them. Bright light late at night delays the whole system. Darkness earlier advances it. The clock is real, but it's also movable, in small increments, by something as ordinary as leaving a lamp on. What the clock does with the information it's now receiving, the long report of sustained dimness, the absence of the short wavelength light that melanopsin is most sensitive to, is begin to release its hold on certain rhythms it has been suppressing all day. One of those rhythms involves the pineal gland and the molecule it produces in darkness. That molecule will come later in this telling. There's a tendency to jump to it because it's the most familiar part of this story, and familiarity makes things feel like conclusions. But the melanopsin cells and the slow clock they feed are not prologue. They are the process itself. The actual biological event and night arriving in the body, before any single hormone has shifted, before any tissue has changed what it's doing. The photon count is dropping. The integrating cells are building their quiet report. The pacemaker is reading something it had been reading every night for your entire life and adjusting in increments too small to feel. The timing of everything that comes next. You've never had to do anything to make this happen. That's the part that tends to land differently when you sit with it. Not the complexity of it, though the complexity is real and strange, but the automaticity. The melanopsin cells don't wait to be told to integrate. The suprachiasmatic nucleus doesn't require your attention to run its program. You could be asleep right now, already, and this system would continue its measurement without you. In a sense, it has been running every night since before you were born. The same proteins, the same pathway, the same slow reading of the dark. Your grandmother's eyes had these cells. The eyes of people long before written language had these cells. The photopigment itself, melanopsin, is deficient enough that versions of it exist in fish, in amphibians, in birds that use it to sense seasonal light changes through the skull. Your retina is carrying something very old tonight, and it is counting the dark patiently without urgency, the way things count that have been counting for a very long time. The body is receiving that count, somewhere downstream of the optic tract, in a structure so small you could hold its entire neuron population in a space roughly the size of a grain of rice. It's reading the report, It's already making decisions you won't feel for another hour. The next ones involve something that has been held back all day, waiting for exactly this signal, coiled quietly in a gland that sits at the almost geometric center of your brain. Something is leaving. Not dramatically, nothing in this chapter announces itself. But if you could measure what's circulating in your bloodstream right now, you'd find less of something than there was at noon. Less than there was at 3 in the afternoon. The slope has been downward for hours already, and it will continue for several more, bottoming out somewhere in the early morning before it begins its slow climb back toward waking. Cortisol, withdrawing. The word carries baggage, stress hormone, people say, and that's not wrong, but it's not the whole picture either. Cortisol is a glucocorticoid produced in the adrenal cortex, released in pulses across the day, highest in the first hour after waking, dropping across the afternoon in most people, and continuing that drop into evening. It is involved in blood sugar regulation, immune modulation, memory consolidation, inflammatory response. It is not purely a stress chemical, it is a timing chemical, a chemical that tells tissues what part of the day it is. When it's high, certain systems run. When it drops, other systems can. That's really all this is. The withdrawal isn't felt as relaxation exactly. That's the thing I keep coming back to. Relaxation implies you are tense, but cortisol's daytime presence isn't tension. It's more like a standing permission for vigilance. A background authorization for all the processes that require you to be, in some operational sense, on. When that authorization begins to be quietly revoked, the body doesn't exhale. It just stops maintaining certain things. Immune cells that were being held at lower activity start running higher. Certain repair processes that cortisol would interrupt or slow find less resistance. The gut, which cortisol influences through enteric nervous system pathways, shifts in its activity patterns. None of this is sudden. There's a phrase in endocrinology, permissive effect. Cortisol is said to have permissive effects on other hormones, meaning it enables their action rather than directly causing it. The withdrawal of cortisol, then, is a withdrawal of permission for some things and the quiet granting of permission for others. The body at night isn't the absence of the day body. It's a different configuration, different things permitted, different things running. The cortisol curve is part of how that configuration gets established, not by adding something new, but by removing something that was keeping other things in check. The part that's easy to miss is how early this starts. By the time you've decided to sleep, cortisol has already been declining for 6 or 7 hours. The preparation happened in the background of the afternoon. While you were doing whatever you were doing, working, sitting, walking through a room, the adrenal glands were already producing less, already beginning the long evening retreat. You were not involved. You didn't authorize it. The suprachiasmatic nucleus, that small pacemaker already described in the chapter before this one, regulates cortisol release partly through its projections to the hypothalamic pituitary adrenal axis. The circadian signal goes in, the cortisol curve comes out, light exposure, meal timing, sleep history, chronic stress, all of these modulate the curve, but the basic shape of it, the rise and fall across a 24-hour period, is driven by the clock. The clock decided hours ago that this was the descending part. It's a short chapter, this one. That feels right. The process it's describing is brief in a certain sense, not brief in duration, because the cortisol curve spans many hours, but brief in drama. There is nothing to observe here. No sensation marks the withdrawal. No feeling tells you that a glucocorticoid is at 60% of its morning peak, or 40%, or the day's low. The body is becoming a different chemical environment incrementally, without asking you to notice. What you might notice, and this is tentative, because interoception varies considerably between people, is something like a small loosening. Not in the muscles, not yet, but in the quality of the hour. An afternoon urgency that no longer quite applies. A sense that the day's logic is losing its grip. Not because you've decided it should, but because something in the circulating chemistry has already moved. The next system to shift is not subtle about it. But cortisol's withdrawal is, it leaves the way a tide retreats. No single moment of departure, just a gradual uncovering of what was always underneath. There is a gland sitting at the geometric near center of your brain that has not seen light directly in the entire course of your life. It once did, or something very like it did. In the ancestors that predate mammals by a considerable distance, in lampreys, in certain lizards, in the tuatara that still exists today almost unchanged from its Triassic form, the pineal gland or its equivalent sits close enough to the skull that light passes through the surrounding tissue and reaches it, directly. The cells respond to photons without any intermediary. There is no retina involved, no neural relay, no hormone acting as messenger. The photon arrives and the gland responds. Your pineal cannot do this. Buried beneath the corpus callosum, shielded by the skull, surrounded by brain tissue that light does not penetrate, it sits in permanent internal darkness, regardless of whether the sun is up or not. And yet it knows. Somehow, through a chain of signals long enough to feel like a game of telephone played across your entire nervous system, it still receives the news. Night has come, or it hasn't. The chain runs like this, though chain makes it sound more deliberate than it is. The melanopsin-containing retinal cells that were described a chapter ago, those slow integrators, counting photon absence across long windows of time, forward their signal along a pathway called the retinohypothalamic tract, arriving at the suprachiasmatic nucleus. The suprachiasmatic nucleus, running its circadian program, sends projections downward through the hypothalamus into the brainstem, continuing through the spinal cord, exiting through the upper thoracic region, travelling up through the superior cervical ganglion, a cluster of sympathetic neurons in the neck, and finally reaching the pineal gland via norepinephrine release onto its cells. The pineal receives a chemical knock on the door. When norepinephrine binds to beta-adrenergic receptors on pinealocytes, the cells that make up most of the gland's tissue, a cascade begins. Cyclic AMP levels rise. Arylalkylamine N-acetyltransferase, the rate-limiting enzyme in melatonin synthesis, becomes active, and the gland begins to convert serotonin, which it has been accumulating during the day, into melatonin. This only happens in darkness, in sufficient sustained darkness. The entire chain, retina to hypothalamus to brainstem to spinal cord to cervical ganglion to pineal, can be interrupted at the retinal level by light. Even a brief bright exposure in the middle of the night suppresses the norepinephrine signal and reduces melatonin production, sometimes for hours afterward. The gland is that sensitive to the upstream message. Melatonin itself is not a sedative. This is worth saying clearly because the supplement exists in doses that vastly exceed what the pineal produces, and the framing around it tends to imply a direct sleep-inducing effect that the endogenous molecule doesn't quite have. What melatonin does is signal. It is a darkness signal, a chemical broadcast that circulates through the bloodstream, reaching tissues and organs across the entire body, telling them not to sleep exactly, but to know that night has come. The suprachiasmatic nucleus has receptors for it and uses the signal to reinforce its own timing. The pituitary receives it. The immune system responds to its presence. Certain peripheral clocks in the gut, in the skin, in metabolically active tissues, They receive melatonin as a kind of timestamp, a broadcast saying this is the dark portion. Adjust accordingly. What does adjusting accordingly mean? It means different things in different tissues. Immune cells shift their activity profiles. Certain metabolic processes slow. Others counterintuitively increase. The gut does different things at night than during the day. And melatonin is partly how it knows which is which. There is something I find difficult to move past when thinking about the pineal, though I want to hold it carefully, without making it carry more weight than the biology supports. For most of the history of thinking about consciousness, from Descartes, who called the pineal the seat of the soul, to the various traditions that have treated it as spiritually significant, there's been a persistent sense that this small gland is somehow central, important in a way that exceeds its mass, and the biology does nothing to discourage that feeling, even if it doesn't support the older claims. The pineal is, in fact, anatomically central in many brains. It is genuinely unique among brain structures in being unpaired, one gland sitting in the midline. It does sit at an unusual anatomical crossroads, and at its function, translating a light signal into a chemical signal the entire body reads, does have a strange grandeur to it when you look at it clearly. But Descartes didn't know about melatonin. He didn't know about the suprachiasmatic nucleus, or the retinohypothalamic tract, or the superior cervical ganglion. What he noticed, perhaps, was the singular nature of the structure, and filled the uncertainty with the largest concept he had available. The biology is stranger and more specific than anything he proposed. Right now, in the darkness, or near darkness you're in, the gland is active in a way it cannot be during the day. The norepinephrine knock arrived some time ago. The enzyme is running. Melatonin is being synthesized at a rate that peaks somewhere in the first half of the night for most people, falling off toward morning, the curve as characteristic as a fingerprint. The molecule is already in your blood, already circulating, already arriving at tissues that have been waiting for it. Not urgently, not dramatically, but the way a room waits for a particular quality of quiet that it only gets at certain hours. There is a strange comfort in the indirectness of it, if you let yourself sit with it. The signal that your pineal is responding to tonight did not originate in the pineal. It came from the back of your eyes, from 3,000 cells counting darkness, forwarded through a relay that spans your entire torso, before arriving at a gland the size of a small grape, buried behind every other structure, in permanent internal night. It has been there your entire life. Receiving the news second-hand, responding as if it had seen the darkness itself, in a sense, through the long chain of proxies that carry the signal it has. The melatonin moves outward from the gland into the bloodstream and then into everything, into the darkness of your gut, into the peripheral quiet of tissues that will spend the next several hours doing work they cannot do while the day's chemistry is running, into the cells that keep their own clocks, loosely synchronized to this signal, adjusting their molecular timekeeping by a few minutes each night. The gland sits in the middle of your brain, in the dark it has always lived in, sending a message about the dark outside. The body, all of it, is listening. And somewhere in the downstream of that signal, in the muscles, in the postural systems that have been holding you upright and ready since you first woke, something that has been performing is beginning very quietly to stop. You've been holding a shape all day, not consciously, that's the thing, you didn't decide to hold it. Nobody does. But the muscles that keep you upright, the ones that brace the jaw and brace the neck and keep the shoulder blades from drifting apart, they've been receiving a steady, low-level signal to stay engaged, to stay ready, to maintain the particular posture that daylight and effort and the presence of other people seems to require. That signal is quieting now, the jaw first usually. Or maybe you notice it in the jaw first, because the jaw is somewhere attention lands easily. The masseter muscle, thick, strong, sitting at the angle of the jaw on both sides, runs almost continuously tighter than it needs to during the day. Not clenching most of the time, just carrying a baseline tone that serves no particular purpose beyond the general maintenance of readiness. The kind of readiness that says, I could speak, I could react, I am, in some operational sense, still on. When cortisol drops, when the arousal signals from the locus coeruleus begin to ease, the motor neurons that sustain that baseline tone reduce their firing rate. Fractionally. Then a little more. The jaw doesn't fall open. It just settles. There's a specific quality to that settling that's hard to describe without sounding like you're describing sleep itself, which hasn't arrived yet. It's more like permission. The muscle discovering it doesn't need to maintain the argument anymore. Beneath the jaw, the neck, the sternocleidomastoid, the deep cervical flexors, the suboccipital muscles tucked at the base of the skull. These have been making small constant adjustments all day, balancing a head that weighs somewhere between 4 and 5 kilograms against the forces of movement and gravity. Even sitting still, these muscles don't rest. They correct continuously tiny adjustments, gamma motor neurons maintaining spindle sensitivity, so the system can respond instantly to any perturbation. That vigilance is calibrated to the day's demands. When the demands withdraw, when the biochemical environment shifts toward night, when melatonin is circulating and cortisol has retreated, and the ascending arousal systems have reduced their broadcast, the gamma motor neuron activity eases, the spindles become slightly less sensitive. The corrections become slightly less frequent. The head doesn't move, but the muscles holding it stop working quite so hard. Then deeper. The erector spinae running along either side of the spine from the sacrum to the base of the skull. The multifidus shorter and deeper, bridging individual vertebrae. These muscles have been sustaining the upright column since you first stood this morning, and they do it so automatically, so far below conscious direction, that most people are genuinely surprised to discover how much effort the body's background machinery puts into something as ordinary as sitting in a chair. They don't release all at once. Nothing in this chapter happens all at once. That's the thing worth holding on to. The change is incremental, distributed across many muscles in overlapping stages, driven not by a single off switch, but by a gradual reduction in the signals that were sustaining the tone. Motor neurons fire less. Muscle fibers recruited for background tension are progressively released from duty. The body's shape softens at its foundation in ways that the body barely registers as change because the change is so slow. There's something almost fond about watching this happen or thinking about it happening because you can't exactly watch it. The body has been performing uprightness all day, not dishonestly but automatically. It is genuinely upright, genuinely functional, genuinely ready. But readiness has a cost, even when the cost is below sensation. The muscles sustaining the performance were spending something, and now in the dark, with the cortisol low and the melatonin moving through the blood and the arousal systems dialing down their signal, the performance is winding up. Not because the body has decided to stop, because the substrate that was sustaining it has changed. The wood of a fire doesn't decide to stop burning. The release goes further than the postural muscles, much further though quietly. Smooth muscle in the blood vessel walls, which has been maintaining vascular tone all day, receives different autonomic input as the sympathetic nervous system reduces its dominance. Vessels in the periphery dilate slightly. This is part of the heat redistribution that will be described more fully in a later chapter. The warmth moving outward to the hands and feet as core temperature begins its nightly descent. The small muscles of the eye, the ciliary muscle that adjusts lens curvature, the muscles controlling pupil diameter, are already responding to the darkness and to the shift in autonomic balance. The eye is doing less. The muscles that serve it are doing less. Even the muscles of the middle ear, which unconsciously tense throughout the day in response to sound, are receiving a quieter signal. This is one of the reasons the world sounds slightly different in the late hours of the night, when you're nearly asleep but not quite. It isn't only that the world has grown quieter, it's that your ear has changed how it's listening. There is a hum from somewhere in the house, or maybe outside, a low, continuous, almost textural sound that sits at the edge of registering. You probably stopped hearing it hours ago, but it's there, and the muscles of your middle ear, softer now than they were at noon, are letting more of its low frequencies through. Your shoulders are lower than they were an hour ago. Your jaw is not quite where it usually lives. The body is returning to something it never entirely left, a baseline beneath the baseline, a resting state that the day's signals continuously interrupted. It isn't sleep. Sleep is its own condition, with its own distinct physiology. But this is what comes just before the threshold, the body quietly laying down the day's shape, releasing the held posture the way you'd set down something heavy you'd been carrying long enough to forget you were carrying it. Somewhere in the brainstem, a nucleus that has been broadcasting wakefulness all day is already reducing its volume, not going silent, just turning down. In the brainstem, roughly at the level of the pons, there is a nucleus so small it contains somewhere between 15,000 and 50,000 neurons depending on the individual. Bilateral, one on each side, mirrored, densely pigmented with neuromelanin, which gives the tissue a bluish colour visible to the naked eye. This is the locus coeruleus. It produces the majority of the norepinephrine that your brain uses, not some of it, most of it. During the day, its neurons fire at a rate of somewhere between 1 and 5 spikes per second, depending on arousal state, task demand, novelty in the environment. When something unexpected happens, a loud sound, a sudden movement, the arrival of information that doesn't match what was predicted, the firing rate jumps. Norepinephrine floods target regions. Attention sharpens. Signal-to-noise ratio across the cortex increases. This is the volume knob going up. The locus coeruleus projects almost everywhere. To the prefrontal cortex, to the hippocampus, to the amygdala, already mentioned in an earlier chapter, to the cerebellum, the spinal cord, the thalamus. Its reach is unusual for such a small structure, a single nucleus, less than a centimetre in length, with axons branching into nearly every major brain region. When it fires, the whole brain gets louder. When it quiets, something like the reverse happens. In the hours after darkness, locus coeruleus firing rate decreases, not suddenly, not uniformly. There are fluctuations, brief increases in response to stimuli, transient spikes that still occur. But the baseline, the tonic firing rate underlying everything else, drifts downward. 1 spike per second instead of 3, then less. Norepinephrine release into target regions decreases proportionally. The prefrontal cortex receives less. The hippocampus receives less. The thalamus, which uses norepinephrine partly to maintain its relay function, forwarding sensory information upward to the cortex, receives less, and its relay becomes less efficient. Sensory information is still arriving at the thalamus. The ears are still working, the skin is still registering contact and temperature, but the thalamic relay is running at a reduced gain, forwarding signals with less fidelity, filtering more aggressively. The world doesn't go away, it loses resolution. There's a particular quality to this that's worth pausing on even briefly. The reduction in locus coeruleus activity doesn't feel like anything specific. It doesn't announce itself. What it produces experientially is harder to locate, a slight flattening of urgency maybe. A reduction in the sense that things require immediate response. The thought that would have completed itself 10 minutes ago now drifts slightly before it finishes. That drift is real. It has a substrate. The prefrontal cortex, receiving less norepinephrine, is running at lower signal gain. Working memory becomes slightly less sticky. The maintenance of task-relevant information, the holding of something in mind against interference, requires more effort than it did an hour ago, and the effort isn't always available. This is not impairment exactly, it's recalibration. The brain is shifting resources away from systems that serve vigilance and towards systems that serve consolidation, restoration, the kind of processing that happens best when the foreground noise has been reduced. The locus coeruleus also projects to the dorsal raphe nucleus, which produces most of the brain serotonin, and to the tuberomammillary nucleus, which produces histamine, another wake-promoting neurotransmitter. As locus coeruleus activity decreases, these regions receive less excitatory input. Their own output decreases in turn. The wake-promoting signal becomes quieter at multiple nodes simultaneously. This is how large-scale state changes work in the brain, not a single off switch. A network of mutually reinforcing reductions, each structure becoming quieter, partly because the structures that were exciting it have become quieter first. The locus coeruleus is not the only node reducing its output tonight, but it may be the first. It's a small structure that keeps coming back. 15,000 neurons at the low estimate, coordinating a signal that reaches into every major region of the brain, modulating attention and arousal and sensory gain and emotional reactivity and memory encoding all at once. The reductions happening in it right now are, in absolute terms, tiny. A few fewer spikes per second, a slight decrease in a neurotransmitter already present in nanomolar concentrations, and yet the effect distributed across the entire brain through those branching projections adds up to something you can almost feel. Almost. The room has not changed. The sounds are the same sounds. The weight of the blanket is the same weight. But the gain on all of it has been turned down, incrementally, by a nucleus smaller than the tip of your smallest finger, sitting in the brainstem you never think about, doing its quiet arithmetic of spikes and silences, less, then a little less. The things that would have pulled your attention 10 minutes ago are losing their grip, not because they've changed, but because the system that assigns urgency to them has become quieter. Upstream, in structures that sit above the brainstem, something else is beginning. The thalamus, already receiving a reduced signal from the locus coeruleus, is beginning a different kind of operation entirely, one that has less to do with forwarding information and more to do with slowly, partially closing the door. You can't see well in the dark. You already know this in the practical sense. The fumbling for a light switch, the strange flat quality of a room at midnight, the way detail dissolves and edges go soft, and colours drain out of everything until the world becomes a study in grey gradients and uncertain shapes. You know this the way you know the feel of your own handwriting, without thinking about it much. But the eye isn't failing in the dark. That's the thing worth sitting with for a moment. It's changing what it's doing. The human retina contains two primary classes of photoreceptor cells: cones, concentrated in the central fovea, optimized for color discrimination and fine detail in bright light, and rods, distributed more densely in the peripheral retina, containing a photopigment called rhodopsin, sensitive to light at levels so low that a fully dark-adapted rod can theoretically respond to a single photon. A single photon, that number tends to pass by without landing. So, in a completely dark room, after 30 or 40 minutes of true dark adaptation, Your rod cells become sensitive enough that a single quantum of light, the smallest possible unit of electromagnetic energy in the visible range, is sufficient to trigger a response. Not enough to see by. You'd need roughly 5 to 7 rods responding simultaneously for the brain to register even a threshold perception. But the cellular machinery is operating at the physical limit of what light detection can mean. This is not primitive technology. This is the opposite of primitive technology. The shift from cone-dominated vision to rod-dominated vision doesn't happen instantly. Dark adaptation is a process, running across roughly 30 to 40 minutes of low-light exposure during which the photopigments that were bleached by the daylight slowly regenerate. Cone pigments recover faster within the first few minutes. Rod pigments, particularly rhodopsin, take longer, but their recovery eventually extends visual sensitivity far beyond what cones can provide in dim conditions. During this window, if you've ever moved from a bright room into genuine darkness, you'll have felt the process running. The first minutes, nearly blind, the world reduced to almost nothing. Then slowly, shapes, outlines, the faint suggestion of a doorframe, a window, the geometry of a room. Not because your eyes adjusted in any casual sense, because rhodopsin was regenerating, rod sensitivity was increasing, and the visual system was recalibrating toward a different operating range. The eye was switching modes. What it switches to is fundamentally different in character from daytime vision. Rod vision, scotopic vision technically, has no colour. Rhodopsin absorbs across a broad spectrum peaked around 498 nanometers in the blue-green range, but the downstream processing doesn't differentiate wavelengths the way cone pathways do. Everything becomes luminance, brightness, and shadow. The world at night, as your rods see it, is monochromatic not because colours aren't present, but because the receptor system you're now using doesn't carry that information, and the resolution drops. Rods feed into a more convergent neural pathway, many rods synapsing onto fewer bipolar cells, feeding into fewer retinal ganglion cells, which increases sensitivity through pooling, but sacrifices the fine spatial detail that cone pathways preserve. You can detect something moving at the periphery in near darkness, but you can't make out its features. The eye in scotopic mode is better at catching movement than reading faces, which is, evolutionarily, exactly the right trade-off. There's something interesting about where the useful vision goes after dark, and this is the part that tends to get overlooked in the practical frustration of not being able to see clearly. During the day, your central vision, the fovea packed with cones, is where the useful information is. You look directly at what you want to see. You point the high-resolution part of the eye at the thing that matters. At night, the fovea is nearly useless. The center of your visual field is the least sensitive region after dark, because there are almost no rods there. What night vision you have lives in the periphery. If you want to see something in genuine darkness, the technique, known to astronomers, known to hunters in every culture that has ever moved through the dark, is to look slightly away from it, to let it fall on the peripheral retina, where the rod density is higher. The useful part of the eye moves after dark from the center outward. I've always found something unexpectedly moving about that. The part of you that sees best in darkness is the part that isn't looking directly. Other animals have refined this further. The tapetum lucidum, the reflective layer behind the retina in cats, dogs, many nocturnal mammals, bounces light back through the rod layer a second time, effectively doubling the chance of photon capture. This is what causes eye shine, the reflected glow when light hits an animal's eye at night. Humans don't have it. We lost it somewhere along the primate lineage, likely as colour vision, requiring cone specialization, became more important than maximal scotopic sensitivity. A trade-off, as most things in biology are. but the rods remained, sensitive enough to detect single photons, distributed across the peripheral retina in a pattern that prioritizes movement detection over detail. Still running the rhodopsin cycle every night, still dark adapting, still operating a system that was essential long before there were rooms to fumble through in the dark. Right now, your rods are doing their work. In whatever darkness surrounds you, at whatever level of light is present, the rhodopsin cycle is running. The eye is not resting. It never quite rests. Even in sleep, retinal processing continues in a reduced form. And the intrinsically photosensitive cells described several chapters ago are still monitoring, still counting, still sending their slow report upstream. The eye has been open to the dark for a long time tonight. Longer than you've been paying attention to it. Since before you decided to sleep, since before the light went off, the rods have been recovering sensitivity in the dim hours of the evening, the peripheral retina becoming gradually more attuned to the low-level light that remained. You can't see well in the dark, but the eye has been doing something in it, continuously, with the patience of a system that has been operating this way across hundreds of millions of years of vertebrate history, looking without quite seeing, attending without finding. Somewhere beneath that patient vigil, in the blood and in the receding chemistry of the day, something is happening to pressure, to the literal pressure of the fluid in your vessels, the tide of force that moves through you with every beat of the heart. It is changing, without your knowledge, without your participation, without any sensation you could name. There are about 120 million rod cells in the human retina, arranged not randomly but in a specific density gradient, sparse at the very center, rising steeply toward a peak density at roughly 20 degrees of eccentricity from the fovea, then declining gradually toward the far periphery. If you could look at a map of your own retina, in terms of rod density, it would look something like a ring, a halo of maximum sensitivity encircling the color-seeing center, a halo of night vision built around the day vision center. The two systems coexist in the same tissue. The cone-heavy fovea, used almost exclusively during daylight, sits ringed by rod-rich peripheral retina that becomes the primary visual system after dark. They share the same neural pathways downstream, the same retinal ganglion cells forwarding signals along the optic nerve, the same visual cortex passing the output. But the information they send at night is fundamentally different in character, lower resolution, achromatic, movement biased, and the brain processes it accordingly. There is a third adaptation worth mentioning, and it lives deeper than the receptors themselves. The pupil, at full dark adaptation, dilates to somewhere between 6 and 8 millimeters in diameter in young adults, compared to roughly 2 to 4 millimeters in bright light. This isn't a minor adjustment. The area of the pupil at maximum dilation is several times larger than at constriction, and since the amount of light entering the eye scales with pupillary area, this dilation alone accounts for a significant increase in retinal illumination. Combined with rhodopsin regeneration in the rods, the total increase in visual sensitivity during dark adaptation spans something like 6 orders of magnitude. A factor of 1,000,000, roughly speaking, between the least sensitive state and the most sensitive. The eye doesn't have one operating range. It has a range of ranges spanning conditions from bright desert noon to starlit overcast night, and it navigates between them through a combination of pupillary adjustment, photopigment regeneration, and neural gain changes that cascade through the retina and into the visual cortex. Most nights you move through the lower portion of this range without noticing it. There are records, in various military and maritime contexts, in the accounts of astronomers who worked before electric lighting, of just how much the dark-adapted eye can perceive under conditions of total natural darkness, away from any artificial light source. Under a clear moonless sky, away from cities, a fully dark-adapted human eye can detect the Andromeda Galaxy, 2.5 million light-years distant, as a faint smear at the periphery of vision. Not because the galaxy is particularly bright, but because the rod system at maximum sensitivity can pull photons from that source across a large enough retinal area to cross the detection threshold. We no longer live in environments dark enough to use this capacity routinely. The dark adaptation that your rods are capable of running tonight is probably not running at its maximum depth, because some ambient light almost certainly remains. A streetlight through a curtain, a standby light on a device, the faint glow of a city sky. True darkness is rarer than it used to be, but the system is still there, still capable of what it was always capable of. The rods that could, under the right conditions, catch the light of a galaxy. They're in the back of your eyes right now, doing what they've always done in the dark, with the same molecular machinery, the same rhodopsin, the same extraordinary sensitivity that the vertebrate eye arrived at across hundreds of millions of years of nocturnal pressure. It's strange to lie in the dark and know this. Or not strange exactly. Strange isn't quite the right word. There's something settling about it, maybe. The eye that can't find the edge of the bedside table in the dark is the same eye that is, right now, running one of the most sensitive light detection systems that biological evolution has produced. The limitation isn't a failure, it's a mismatch, a system designed for genuine, deep, pre-electric darkness, operating in the residual glow of a world that never fully goes dark anymore. The rods are working with what they have, they always have, and they will continue through the night, through whatever quality of dark surrounds you, regenerating rhodopsin in cycles that run whether or not you're awake, maintaining the machinery of night vision in a body that is increasingly preparing to stop needing it until morning. The pupil, wide now, admits whatever faint light remains, the peripheral retina attends, and the rest of the body, the pressure systems, the fluid mechanics, the quiet cardiovascular adjustments that have been running since evening without a single sensation to mark them, continues its own patient unannounced work. Tonight, sometime in the next few hours, your blood pressure will drop. Not because of sleep necessarily, not because you've relaxed or decided something or done anything at all. It will drop because it drops every night, on a schedule as reliable as the melatonin curve, driven by the same circadian machinery that has been running since long before you had any say in the matter. The drop is called nocturnal dipping. Most people have never heard the term. In clinical settings, blood pressure is measured at a single moment, a cuff, a number, a comparison to a range. But blood pressure is not a fixed value. It moves continuously across the day and night, in response to posture and activity and stress and digestion and a dozen other variables. The circadian component of this movement is one of the most consistent patterns in human cardiovascular physiology. Pressure rises in the early morning hours before waking, peaks somewhere in the mid-morning, declines across the afternoon, and then in the hours after darkness and into sleep drops further, typically by 10 to 20% in people whose cardiovascular systems are functioning within a normal range, 10 to 20% every night, without a single sensation to mark it. The mechanism runs through the autonomic nervous system. During the day, sympathetic tone, the branch of the autonomic system associated with arousal and readiness, maintains a baseline level of vascular resistance. The smooth muscle in arterial walls receives continuous low-level signals to contract, keeping the vessels narrower than they would be at rest. The heart rate is sustained at a slightly higher baseline. The whole system is tuned for a body that might need to respond. At night, sympathetic tone decreases. Parasympathetic activity increases. The smooth muscle in vessel walls receives less signal to contract and relaxes fractionally. The peripheral resistance drops. The heart rate slows slightly. Blood pressure, which is fundamentally a product of cardiac output, and vascular resistance falls. The heart is doing the same work it always does. The pipes, briefly, have widened. There is a category in cardiology, non-dippers, the term is, for people whose blood pressure does not follow this nocturnal pattern. Non-dipping is associated with increased cardiovascular risk, with disrupted sleep architecture, with certain kidney conditions and autonomic dysfunction. The research on this is reasonably consistent and has been accumulating for several decades. What that research implies, without quite stating it outright, is that the nocturnal dip isn't incidental. It isn't a side effect of lying down or of reduced activity. It is a designed feature of the cardiovascular system, a nightly recalibration that the body appears to need, that it has been performing reliably for the duration of human evolutionary history, and that its absence correlates with things going wrong. The dip matters, and it asks nothing of you to run, right now, whether you're nearly asleep or still hovering at the surface of wakefulness. The autonomic shift is already underway, has been for a while. The sympathetic nervous system began its evening retreat hours ago as part of the same circadian cascade that lowered cortisol and slowed the locus coeruleus and directed the pineal to produce melatonin. These systems don't operate in isolation. They are coupled, entrained to the same central clock, declining together in a pattern that the body coordinates across multiple organ systems simultaneously. Your blood pressure right now is lower than it was at noon. You haven't felt any of this. That's the part worth sitting with, even briefly, not because it's alarming, it isn't, but because it's a reminder of how much of the body's life runs entirely outside experience. The cardiovascular system is adjusting pressure across your entire vascular network, from the large arteries near the heart to the small arterioles in your fingertips and the capillaries in the skin behind your ear, and none of it produces a signal that reaches consciousness. No sensation of vessels widening, no awareness of the heart rate easing, no feeling of the pressure dropping, just the body doing it, as it does every night, as it has done every night of your life in the background of every sleep you've ever had, unnoticed and unannounced and entirely reliable. The house settles around it. The body continues. And somewhere in the immune tissue, in the lymph nodes and the circulating cells that have been waiting all day for the body's attention to turn inward, something is beginning to stir. There is a version of this that gets told dramatically. The immune system as an army, night as the time when the soldiers finally get to work, the body defended while you sleep. That version isn't entirely wrong, but it flattens something. It makes the immune system sound like it was waiting to be heroic. What it was actually doing was waiting for the cortisol to drop. Cortisol suppresses immune activity. This is not incidental to its function, it's one of its primary roles. The glucocorticoid receptors on immune cells respond to cortisol by down-regulating the production of pro-inflammatory cytokines, reducing the proliferation of lymphocytes, dampening the kind of immune activity that, if it ran continuously, would be costly to maintain and potentially damaging to the body's own tissues. During the day, when cortisol is high, the immune system runs at a lower operational level. Not inactive, but held. As cortisol retreats across the evening, that long, quiet withdrawal described a few chapters back, and the immune system finds less resistance. The glucocorticoid break eases. Cell populations that were being held at reduced activity begin to increase their output. Not because a signal told them to start, but because the signal that was telling them to stay quiet has become weaker. This is the immune system's night, beginning not with an activation but with a release. The building empties differently than it fills. During the day, the corridors are full, foot traffic, noise, the constant interruption of whatever low-level maintenance work needs doing. At night, after the last person has left and the hum of activity has settled, the building reveals a different set of workers. Ones who were there all along, running quieter operations, waiting for the space to do them. The immune system at night is something like that, not transformed, just given room. Natural killer cells are among the first to show a circadian pattern that favours the dark hours. These are lymphocytes, a type of white blood cell that patrol for cells displaying signs of viral infection or malignant transformation and eliminate them through direct cytotoxic activity. Their circulating numbers and functional activity follow a daily rhythm, with peaks in the late evening and early night in humans. The mechanisms driving this rhythm involve both the direct effects of melatonin, which has receptors on NK cells and appears to enhance their activity, and the withdrawal of cortisol's suppressive influence. NK cell activity at night isn't a surge, it's a return to a higher baseline. The daytime level was the suppressed state. T cells follow a different pattern, and this is where the circadian immunology becomes genuinely strange if you let yourself think about it carefully. T cells, the adaptive immune system's most targeted workers, capable of recognizing specific pathogens and coordinating responses against them, migrate. They move between the blood and the lymph nodes in a pattern that follows the circadian clock. During the day, more T cells circulate in the blood, available for rapid deployment if a pathogen is encountered. At night, T cell trafficking into lymph nodes increases. More cells move into the lymphoid tissue, where they can interact with antigen-presenting cells, receive information about what the immune system has encountered recently, and potentially be primed for future responses. The lymph node at night is busier than the lymph node during the day. This is not what most people would guess. The intuition that the immune system is more active and more alert during waking hours, when you're more likely to encounter pathogens, is reasonable but apparently incorrect, at least in this specific regard. The preparation happens at night. The cataloguing, the cross-referencing, the slow work of immune memory, this runs preferentially in the dark hours. It's been doing this your entire life, in lymph nodes you've never thought about, in tissue tucked into your neck and your armpits and along your gut wall, quiet and unhurried. Cytokines, the signaling molecules that immune cells use to communicate with each other and with other body systems, also follow circadian rhythms that favour nighttime expression for certain members of the family. Interleukin-2, which drives T-cell proliferation. Interleukin-12, involved in natural killer cell activation. Tumour necrosis factor. These are not uniformly elevated at night. The picture is more specific than that, with different cytokines peaking at different points in the circadian cycle. But the overall pattern, across multiple studies and the multiple cell types, suggests that the immune system's most active coordination work happens during the biological night. Sleep deprivation disrupts this, not metaphorically, measurably. Even a single night of significantly shortened sleep alters cytokine profiles, reduces NK cell activity, and appears to impair the T-cell trafficking into lymph nodes that normally occurs during the dark hours. The immune system doesn't catch up easily. Some of what was missed, in terms of immune coordination and maintenance, simply doesn't happen when the window closes. This is not said as a warning, just as a fact about timing. There is something worth noticing about how all of this fits together, even if fitting together makes it sound neater than it is. The same circadian cascade that lowers cortisol and slows the locus coeruleus and directs the pineal to produce melatonin, the same central clock, the same chain of signals, also coordinates the immune system's nightly shift. Melatonin itself acts on immune cells directly. The reduced sympathetic tone that allows blood pressure to dip also reduces the adrenergic signaling that suppresses certain immune functions during the day. These are not separate systems running separate programs, they are coupled. The body's nighttime reconfiguration happens across multiple organ systems simultaneously, coordinated through overlapping hormonal and neural signals that are all ultimately entrained to the same light-sensitive pacemaker sitting above the optic chiasm. The immune system's night begins when the eye's slow-counting cells report that the dark has been present long enough. Right now in the lymph nodes you'll never feel small, dense, bean-shaped structures distributed throughout your body like quiet switching stations. T cells are moving, arriving through the high endothelial venules that serve as their entry points, settling into the cortex of the lymph node, making contact with dendritic cells that have spent the day collecting antigenic information from the body's periphery. There is no sensation for any of this. There is no way to feel a T cell entering a lymph node. The body performing this work is entirely silent from the inside, but the work is happening. It has been happening every night of your life. The immune system, given the dark and the lowered cortisol and the melatonin moving through the blood, doing the slow, unglamorous maintenance work that keeps you not dramatically, not heroically, but steadily, reliably, night after night, intact. Somewhere in the body's interior, in tissue that runs along your gut wall and sits behind your sternum and clusters in the soft places of your neck, a kind of quiet congress is underway. And above it, in the brain, in the place where the day's accumulated noise of being a thinking, sensing, remembering creature begins to find less and less to hold on to, something is turning inward, not towards sleep yet, toward itself. You may have noticed it already, not heard it, or not exactly, more like become aware of it. A low rhythmic presence somewhere in the chest or throat or the soft tissue behind the sternum, a pulse that has been running continuously since before you were born, and that you almost certainly haven't thought about since sometime earlier today when you were doing something else entirely. It didn't get louder. The room got quieter. This is interoception, the body's sense of its own interior state, and it is one of the less discussed sensory systems, possibly because it doesn't produce the kind of vivid, locatable experiences that vision and hearing do. It produces instead a background hum of bodily self-knowledge, the awareness of fullness or hunger, the felt sense of a breath, the vague pressure of a bladder not yet urgent, the heartbeat noticed in stillness. These signals are always being generated. The body is continuously reporting on itself through dedicated nerve pathways to dedicated brain regions. What changes at night, in the quiet, in the low light, in the reduced sensory traffic of a body settling towards sleep, is the competition. During the day, external sensory input is high. Vision, hearing, touch, proprioception, the sense of your body's position in space, all of these generate continuous neural traffic moving toward the cortex, competing for processing resources, occupying the attentional foreground. Interoceptive signals are present in this environment, but they're relatively weak against the noise. The heartbeat is there, the gut is reporting, the breathing pattern is always generating data, but most of it doesn't reach consciousness because it doesn't need to, because the external world is louder, more immediately relevant, more demanding of response. In the dark, lying still, with external input reduced, and the arousal systems quieting and the sensory relay in the thalamus running at lower gain, the ratio shifts, the external noise drops. The internal signal, unchanged, suddenly has less to compete with. The heartbeat you can feel right now, or might feel if you let your attention move toward the chest, is traveling toward your brain along several pathways simultaneously. The vagus nerve carries much of it. The vagus is the 10th cranial nerve, the longest of them, running from the brainstem down through the neck and chest and into the abdomen, branching as it goes, innervating the heart and lungs and most of the digestive tract. Its name comes from the Latin for wandering. It does wander. It touches more organs than any other single nerve in the body. Most of what it carries is travelling upward, afferent signals moving from the body's interior toward the brain, reporting on the state of organs and vessels and tissues that the brain has no other way of knowing about directly. The heart's rhythm, the stretch of the lungs, the state of the gut wall. This information arrives in the brainstem first at the nucleus of the solitary tract, then moves to the thalamus and from there to several cortical regions, most notably the insula. The insula, it sits folded inside the lateral sulcus of the brain, tucked between the frontal and temporal lobes, invisible from the outside of the brain. Its posterior portion is where interoceptive signals from the vagus and other pathways arrive and are initially processed, where the heart rate becomes a felt sensation, where the breathing becomes something you can attend to rather than just something that happens, where the body's continuous self-report is translated into experience. The anterior insula is involved in the more interpreted, emotionally inflected version of that experience. The feeling of anxiety, the felt sense of embarrassment, the way physical illness has an emotional quality as well as a physical one. The two regions talk to each other. The raw report from the body becomes, by the time it reaches the anterior insula, something with emotional colour. The heartbeat you feel right now is not just mechanical information. By the time it arrives in the insula, it has been processed, integrated, and given a felt quality. Right now that quality is probably quiet, a little slow, steady in a way that daytime pulses rarely feel steady, because the context around it has changed. There is individual variation in how vividly people experience this. Some people are highly interoceptive. They notice their heartbeat easily, feel their breathing without effort, have a clear and continuous sense of their body's interior. Others find the same signals faint, difficult to locate, almost absent from awareness even in stillness. This variation is real and measurable, and it seems to involve both the sensitivity of the vagal afferents themselves and the degree to which the insula processes and represents their signals. Neither extreme is inherently better. High interoceptive sensitivity is associated with more vivid emotional experience, but also sometimes with heightened anxiety. Lower sensitivity has its own qualities. But even people who rarely notice their heartbeat during the day often find it in the dark, in the quiet. When the external competition has dropped enough, the signal was always there. The window opened. I find myself thinking about this more than it probably warrants. The fact that the body has been talking to itself continuously, heart to vagus, to brainstem, to insula, to consciousness, every moment of every day, and that most of this conversation happens below awareness, and that it only surfaces when the external world gets quiet enough to let it through. There's something in that which feels important, though I'm not sure important is quite the right word. Maybe just worth knowing. The body has an interior life in a purely biological sense that runs independent of what you're paying attention to. The insula is receiving the heart's report right now. It has been all day. You just couldn't hear it over everything else. Your heartbeat is probably slower than it was this morning. The locus coeruleus has reduced its norepinephrine output. The parasympathetic nervous system has increased its relative dominance. Heart rate variability, the subtle beat-to-beat variation in timing that reflects autonomic balance, is likely higher than it was at noon, which is counterintuitively a sign of the system working well. A perfectly regular heartbeat, with no variation at all, would suggest reduced autonomic flexibility. The slight irregularity of a resting parasympathetically dominant heart is the healthy kind of irregularity, each beat varying fractionally from the last, each variation a small report on the state of the whole system. The pulse in the chest, the faint awareness of breathing, the slight rise and fall that you might or might not be tracking, the body's temperature gradient, warmth collecting in the hands. These are all interoceptive signals arriving at the insula right now, building the brain's current model of what the interior of the body feels like. That model, at this hour in this quiet, is probably the softest version of it you've encountered all day. The external world has stopped competing. The body's self-report is arriving into less noise than it has had access to since you woke. And deeper than the insula, deeper than the felt heartbeat, deeper than the vagus and the brainstem and the circulating melatonin, in the tissue of the skin at the surface where this whole chapter of the night began, temperature is doing something it does every night, something tidal and slow and completely beyond decision, moving. Your hands are warmer than they were an hour ago. You may not have noticed. Warmth that arrives gradually without contrast tends not to announce itself. It simply becomes the new condition, and the previous condition is forgotten as quickly as the shift was made. But if you paid attention to the hands right now, to the specific quality of their warmth, to the way the skin feels slightly fuller, slightly more present than it does in the middle of the afternoon, that change is real. And it has a precise biological source. The body is moving heat, not generating more of it, not losing it randomly to the air around you, but actively redistributing it, pulling it from the core and directing it toward the periphery, toward the skin of the hands and feet and face where it can be released into the surrounding air and carried away. This is not an accident of lying still. It is a coordinated physiological event, timed to the circadian clock serving a specific and necessary function. The body needs to cool its core. The preoptic area of the hypothalamus, a small region sitting just anterior to the optic chiasm, close to the suprachiasmatic nucleus that has already appeared several times in this telling, contains the brain's primary thermoregulatory center. Warm sensitive neurons in this region respond to increases in blood temperature by initiating heat dissipation responses. Cool sensitive neurons do the reverse. The preoptic area is, in a sense, the body's internal thermostat, though that word makes it sound simpler and more mechanical than it actually is. What the preoptic area does at night is different from what it does during the day. Under circadian influence, receiving input from the suprachiasmatic nucleus, integrating the hormonal signals of the evening, it shifts its set point downward, not dramatically. Somewhere between 0.5 and 1 degree Celsius over the course of the night, in most people. The core temperature that the body was maintaining at 37 degrees, or close to it during the day, is no longer the target. The target becomes slightly lower, and the body begins working to reach it. The mechanism is peripheral vasodilation, the smooth muscle in the walls of the blood vessels supplying the skin, particularly the cutaneous vessels of the hands, feet and face, relaxes. The vessels widen. More blood moves through the peripheral circulation, bringing core warmth to the surface, where it radiates and convects into the cooler air of the room. The skin over these regions flushes slightly, though not enough to be visible in ordinary light. The surface temperature of the hands and feet rises. The core, losing heat to the periphery, begins its slow descent. This is the hands warming as the core cools. The two movements are the same event, seen from different locations in the body. There is something in this that tends to land differently when you encounter it at night, lying still, with the biology actually running around you rather than being described in the abstract. The warmth in the hands right now is not comfort in the usual sense, not warmth from a cup or a fire or proximity to another person. It is the body's own heat being deliberately moved outward, routed through the peripheral circulation to the surface given to the air. The hands are warm because the core is releasing something. The warmth you might feel there, if you press one palm against the back of the other hand, is heat that was minutes ago, somewhere near the centre of the body. It has travelled to meet the air. The ground does something like this after dark, releasing the warmth it accumulated during the day, slowly into the night sky, cooling from below as the heat moves upward and away. The body does something similar, though more deliberately, more precisely, through specific vascular channels rather than through the slow conduction of soil. The metaphor holds for a while and then doesn't. The body is more controlled than the ground. The vasodilation is regulated, reversible, responsive to feedback. It isn't simply releasing heat passively, it is actively managing a temperature gradient toward a specific biological target. But the directional quality of it, the warmth moving outward while the center cools, that part is the same. Why does the core need to cool? The relationship between core temperature and sleep onset has been studied carefully enough that the pattern is clear, even if the full mechanistic explanation still has open questions. Sleep onset is strongly associated with the descending phase of the core temperature rhythm, the period when the body is actively cooling, artificially warming the feet and hands, using warm socks, warm water immersion before bed, has been shown in several studies to accelerate sleep onset, apparently because it enhances the peripheral heat loss that drives core cooling. The body, in other words, cannot easily enter sleep while core temperature is still high or rising. The cooling is not a consequence of sleep, it precedes it. It is one of the conditions that sleep requires. The preoptic area, managing the thermoregulatory setpoint shift, is preparing a specific internal environment, and sleep onset appears to be gated, at least partly, on that preparation being sufficiently complete. This is why sleeping in a room that is too warm often produces fragmented, shallow sleep. The environment is competing with the body's attempt to lose heat through the skin, keeping core temperature higher than the descending set point is trying to reach. The body has a temperature it needs to be at, and it is trying right now to get there. The feet, if you pay attention to them, really attend to their weight, their particular warmth, the way they press into whatever surface they're resting on, are doing the same thing the hands are doing. Cutaneous vasodilation in the plantar surface, in the toes, in the skin over the ankle. Heat moving through the peripheral circulation to the surface, releasing into the air or into the bedding, contributing to the slow core temperature descent that has been underway since the evening began. There is a weight to warm feet that cold feet don't have, a specific settled quality. Feet that have been cold tend to feel slightly remote, distant from the rest of the body in some hard-to-describe way. Warm feet feel present, part of the whole. This is not fanciful. The circulation running through warm feet is genuinely fuller than the circulation running through cold vasoconstricted feet. More blood, more warmth, more metabolic activity in the tissue. The presence is real. The core temperature will continue to fall across the night, reaching its minimum sometime in the early morning hours, typically around 4 or 5 in the morning in most people, before beginning its slow rise back toward the waking baseline that will peak, as it always does, in the late afternoon. The rhythm is as consistent as any circadian pattern the body produces. Right now the descent is in its early hours. The hands are warm, the feet have settled. The blood moving through the peripheral circulation is carrying the day's last warmth outward. giving it to the air with a generosity that requires no decision and no effort, only the quiet opening of vessels that the hypothalamus has already arranged. The room receives it. The body, at its centre, grows fractionally cooler. And in the brain, in the tissue that has been managing all of this, the temperature set points and the vasodilation and the melatonin and the immune trafficking and the blood pressure and the locus coeruleus and the slow retinal count of the dark, something that has been watching over the day's entire operation is preparing to hand that operation over to a different set of systems entirely. Old systems, older in some cases, than the structures that manage waking. Patient ones. In the dark, the iris opens. Not quickly. This isn't the fast, startle-driven response to a sudden change in light that happens in milliseconds, a brisk constriction or adhesion driven by the pupillary light reflex running through the midbrain. What happens in sustained darkness is slower, more considered, if a reflex can be considered. The dilator pupillar muscle, a thin radial sheet of smooth muscle cells running outward from the pupil's edge like spokes, receiving sympathetic innervation, contracting slowly when the norepinephrine signal arrives, pulls the pupil wider across minutes, not moments. 5 millimeters, 6, sometimes 8 in younger eyes, in genuine darkness after enough time has passed. The sphincter pupillae, the circular muscle that constricts the pupil in bright light, relaxes, steps back. The dilator takes the aperture and slowly, without announcing it, makes it larger. Sharks do this. The pupils of certain shark species dilate in low-light conditions, the iris responding to the same basic principle: more aperture, more photons, better chance of detection, through a mechanism that looks, in its broad outlines, similar to your own. Frogs do it. Cats do it with particular drama, the pupils going from narrow vertical slits in daylight to wide dark circles in dim conditions. The cephalopods, octopuses, cuttlefish, have pupils that change shape in ways mammalian eyes don't, but the underlying drive is the same. The dilation of the pupil in darkness is one of the most conserved behaviors in animal vision, which means your iris, opening right now in whatever low light surrounds you, is doing something that connects you, through an unbroken chain of inherited reflex, to eyes that predate your lineage by an almost unimaginable distance. The strange part, and this is the part worth sitting with, is what the dilation actually accomplishes in your particular situation tonight. Probably not much. If the room is genuinely dark, there is little additional light to gather. A wider pupil in near total darkness admits more of almost nothing. The rods, already described in an earlier chapter, are doing their own slow sensitivity adjustment through rhodopsin regeneration. The pupillary dilation is a coarser, faster tool, useful for the transition from moderate to low light, but offering limited benefit in true darkness. And yet the iris opens anyway. The reflex doesn't check whether there's anything worth seeing. It doesn't evaluate the ambient light level and decide whether dilation is warranted. The sustained absence of light triggers the sympathetic signal. The sympathetic signal reaches the dilator pupillae, and the muscle contracts, slowly, steadily, regardless of whether the expanded aperture will be useful. Preparation without verification. That's the operational logic here. The reflex assumes, without checking, that if darkness is present, there may be something to see, that the darkness is a condition requiring readiness rather than a condition requiring rest. It is a very old assumption, running in tissue that has been making it far longer than any organism has been able to evaluate whether the assumption is correct. In the environments where this reflex was shaped, genuine, deep, predator-present darkness, before electric light, before enclosed shelter, before any of the conditions that make nighttime safe in the way it currently is, the assumption was probably correct often enough to matter. An eye that dilated in darkness might catch the movement that an unconstricted eye would miss. The few extra photons gathered by a wider aperture might, on the right night, be the difference between detecting something and not. That logic no longer applies to your bedroom. The reflex applies anyway. There is something I keep returning to about this, quietly, without wanting to overstate it. The reflex is anticipatory in a way that most biological processes aren't. Most of what the body does at night is responsive. Cortisol drops in response to the circadian signal. Melatonin rises in response to the absence of retinal light input. Blood pressure falls in response to reduced sympathetic tone. These are responses. The pupillary dilation is also a response, technically, to the absence of light, mediated by the sympathetic nervous system. But its purpose is oriented toward what hasn't happened yet. It is opening to possibility, preparing for something that may not arrive. The iris doesn't know there's nothing there. It prepares anyway. I find that on certain nights, almost moving. The dilator pupillae is not a large muscle. A thin radial sheet, perhaps 0.5 millimeter thick in the stromal layer of the iris, smooth muscle cells arranged in a wheel pattern, each one tiny, each one receiving its sympathetic signal from the long ciliary nerves, branching ultimately from the superior cervical ganglion, the same ganglion, incidentally, that delivers the norepinephrine signal to the pineal gland that initiates melatonin synthesis. The same sympathetic pathway serves both. The pineal receives its darkness signal and begins producing melatonin. The iris receives its darkness signal and begins dilating. Two organs, both responding to the same upstream message about the same environmental condition, doing different things with it. One chemical, one mechanical, through branches of the same nerve. The body's nighttime responses are more coordinated than they appear from the outside, or from the inside for that matter. Right now, the iris has probably been open for a while. The dilation happens early in the dark adaptation process, preceding the full rhodopsin regeneration in the rods by a significant margin. By the time you're reading this, hearing this, the mechanical adjustment has long since completed. The pupil has found its dark adapted diameter and is holding it. Holding it for what? Nothing, probably. The room is what the room is. The dark is not concealing anything that the wider aperture will reveal, but the eye is ready, in the way that something can be ready for a situation it will not encounter. The iris opened in the dark because that is what irises do, because that is what they have always done, because the muscle and the nerve and the signal and the reflex are still running the program that was written in a much older world, faithfully, without revision, in the back of your eye right now. The oldest reflex, still running. The body that carries it is very nearly asleep, and somewhere deeper than the iris, deeper than the retina, deeper than the optic nerve and the lateral geniculate and the visual cortex, processing the near nothing that the dark provides. In the brain's white matter, in the axons that connect one region to another, in the spaces between neurons that expand slightly in the night, something is being cleaned, something that accumulates all day and cannot be cleared until now. For most of the history of neuroscience, the brain was understood primarily through its neurons. This is understandable. Neurons are dramatic, they fire. They produce action potentials with measurable voltages. They connect in networks of staggering complexity, and those connections change with experience and learning and memory. Neurons are the story that neuroscience told itself for a long time, and the story wasn't wrong, it was just incomplete. The brain contains roughly as many glial cells as neurons. Some estimates put the ratio higher, and glial cells, for most of that history, were described as support cells, the scaffold, the insulation, the maintenance crew for the real performers. The maintenance crew, it turns out, was doing something extraordinary. Astrocytes are the most numerous glial cell type in the brain. Star-shaped, the name comes from the Greek for star. They extend long processes that contact neurons at synapses, wrap around blood vessels, tile the brain's tissue in an almost continuous network of interconnected cells. They regulate the extracellular environment, controlling ion concentrations, removing excess neurotransmitters from synaptic clefts, supplying neurons with metabolic substrates. They are, in the most literal sense, the brain's housekeeping system. But astrocytes have a specific nighttime function that was not clearly understood until the early 2000s, when a series of experiments in mouse models, and subsequent research extending toward human biology, revealed something about the sleeping brain that nobody had quite expected. The brain cleans itself, not metaphorically, through an actual fluid clearance system driven by the flow of cerebrospinal fluid through channels formed by astrocytic processes, flushing the interstitial space of the brain, the narrow gaps between cells, of the metabolic waste products that accumulate during waking neural activity. The system is called the glymphatic system, a portmanteau of glial and lymphatic, reflecting the astrocytes' central role and the functional parallel with the body's lymphatic drainage elsewhere. Its mechanism relies on aquaporin 4, a water channel protein expressed at high density on the end feet of astrocytes. The specialized processes that wrap around blood vessels throughout the brain. Cerebrospinal fluid produced in the choroid plexus of the ventricles flows along the outside of cerebral arteries, entering the brain tissue through the perivascular spaces that surround them. From there it moves through the interstitial space, the fluid-filled gaps between neurons and glia, driven partly by arterial pulsation, partly by the pressure gradients created by this movement, and eventually draining out along venous vessels toward the cervical lymph nodes. Along this route it carries waste. What waste? The metabolic byproducts of neural activity, of neurons firing, of synapses transmitting, of the brain doing the continuous energy-intensive work of being awake and processing the world. Among these byproducts is amyloid beta, a peptide produced as a normal consequence of neuronal metabolism, present in small concentrations in all active brains. In the awake brain, amyloid beta accumulates in the interstitial space across the day. In the sleeping brain, particularly in the deeper stages of sleep, the glymphatic system clears it, not dramatically, not completely in a single night, but measurably. Studies using imaging techniques that can track cerebrospinal fluid flow and interstitial solute concentrations have shown that amyloid beta clearance rates are significantly higher during sleep than during waking, and that glymphatic flow itself is substantially more active during sleep. The brain, in the dark, in the quiet of reduced arousal, is doing something with the space between its cells that it cannot do when the arousal systems are running high. Here is the part that tends to stop people. The interstitial space of the brain, the gaps between cells, expands during sleep by a measurable amount. In the mouse studies where this was first characterized clearly, the interstitial space increased by roughly 60% during sleep compared to waking states. The cells themselves don't shrink exactly. What happens is more complex, involving changes in cell volume regulation, in the osmotic behavior of astrocytes, in the fluid dynamics of the tissue. But the net effect is that the sleeping brain has more room between its cells. It creates space inside itself, and through that expanded space, cerebrospinal fluid moves more freely, carrying more waste out of the tissue, clearing the interstitial environment more efficiently than the waking brain, with its compressed interstitial space and its high arousal signals, can manage. The fluid is moving, slowly without sensation, without any signal reaching consciousness. The brain is using the quiet to do what the quiet makes possible. There is particular quality to thinking about this at the end of the day. The brain that has been working since waking, processing, deciding, remembering, worrying, noticing, constructing continuous experience out of the raw material of sensation and thought, is now, in the dark, in the reduced arousal of the late hours, beginning to process itself. Not its contents, its substrate, the physical material of the tissue, the fluid environment between cells, the chemical residue of the day's activity. The neurons that fired all day left something behind. The astrocytes are moving through the space between them now, in their patient, unhurried way, clearing what was left. Not urgently, not dramatically, the way maintenance happens when the building is finally empty and there is room to work. Microglia, the brain's immune cells, distinct from astrocytes, also glial, are more active during sleep as well, extending their processes, surveying synapses, performing the pruning and monitoring that contributes to synaptic maintenance and plasticity. The brain at night is attended by cells that were present all day but doing different things. The same tissue, different operations. The glymphatic flow will run through the night, peaking during the slow wave sleep that tends to dominate the earlier part of the night's sleep architecture. The amyloid beta that accumulated across the day will be reduced. Other waste solutes, tau protein, metabolic byproducts, ions that shifted during neural activity will be cleared or rebalanced. The interstitial space will contract again toward morning as arousal returns and the physiological conditions that permitted the expansion reverse. The brain will wake with cleaner fluid around its cells than it had at midnight. It will not know this. There is no sensation for it, no experience of having been cleaned, no awareness of the clearing that happened while consciousness was elsewhere, but the tissue will be different. And in the molecular clocks running inside that tissue, inside almost every cell, not just the neurons and glia, but the liver and the skin and the gut wall and the circulating immune cells, a timing system older than the brain itself is running a program that has nothing to do with consciousness and everything to do with time, keeping it. Across every cell in the body simultaneously, faithfully, in a language made of proteins. Inside almost every cell in your body something is counting. Not a neuron, not a structure you could point to on an anatomy diagram, something smaller, a loop of gene expression and protein degradation, running on a roughly 24-hour cycle, present in liver cells and skin cells and immune cells, and the cells lining your gut wall, and the cells in your heart muscle, and the cells in the tissue behind your eyes, each one keeping its own time. The molecular clock runs through a feedback loop. Clock and BMAL1 are transcription factors, proteins that bind to DNA and activate the expression of target genes. Among the genes they activate are period genes, PER1, PER2, PER3, and cryptochrome genes, Cry1, Cry2. The period and cryptochrome proteins accumulate in the cytoplasm across several hours, then enter the nucleus together and inhibit the very clock BMAL1 complex that produced them. The loop closes on itself. The inhibition reduces PER and Cry production. The existing proteins are gradually phosphorylated and degraded. The inhibition lifts. Clock and BMAL1 become active again. PER and Cry begin accumulating again. One cycle of this takes approximately 24 hours. Not exactly 24 hours. The endogenous period varies between individuals, which is why some people naturally run slightly fast or slightly slow relative to the solar day. But close enough that the external light-dark cycle, received through the retina and the suprachiasmatic nucleus, can entrain it, nudging it daily into alignment with the actual rotation of the Earth. The part that takes a moment to absorb is the distribution. The clock in your liver is not the same clock as the clock in your skin. They're running the same molecular loop, the same proteins, the same feedback structure, but they're running it in their own cells, independently, without direct communication with each other. The liver's clock can drift out of phase with the skin's clock if their entrainment signals diverge. Eating at unusual hours, for instance, can shift the liver's molecular clock relative to the light-entrained central clock. The liver takes its timing cues partly from metabolic signals like glucose and insulin, and a meal in the middle of the night sends a very different signal to the liver's purr and cry genes than a meal at noon. The body, in other words, is not one clock. It is many clocks, loosely coupled to each other, each cell running its own molecular loop, coordinated imperfectly through the hormonal and neural signals that the suprachiasmatic nucleus broadcasts outward. Melatonin is one of those broadcast signals. Cortisol is another. Body temperature rhythm is a third. The nightly core temperature drop described earlier in this telling reaches the peripheral tissues as a thermal signal that some molecular clocks use as an entrainment cue. Right now, in the darkness and the late hour, the molecular clocks in your cells are at a specific phase of their cycle. The purr and cry proteins in most of your tissues are likely in the accumulation phase, building up toward the nuclear translocation that will close the loop in a few hours. The clock BMAL1 complex is active in many tissues, driving the expression of clock-controlled genes, genes involved in metabolism, in cell division, in DNA repair, in immune function, that are timed to run at specific phases of the circadian cycle. Your cells know what time it is, not in any experienced sense, not consciously, but the molecular machinery inside them is in a state that corresponds to this hour, to this phase of the biological night, to this particular point in the daily loop that has been running since before you were born and will run until the body stops running entirely. The skin's clock is interesting and underappreciated. Keratinocytes, the cells that make up most of the skin's outer layer, run robust circadian clocks that regulate DNA repair, cell proliferation, and barrier function. UV-induced DNA damage, which occurs during daylight, is repaired more efficiently at certain phases of the circadian cycle. Skin cell division peaks at night in most organisms during the phases when the proliferating cells are less likely to be exposed to UV radiation. The clock in the skin coordinates the skin's biology with the predictable environmental cycle of light and dark, running a program that anticipates the day's damage before it arrives. There are clock genes expressed in hair follicles, in adipose tissue. In the adrenal cortex, where cortisol is produced, the adrenal clock helps shape the daily cortisol curve, and disrupting it disrupts the rhythm. In the retina itself, where local clocks modulate photoreceptor sensitivity in ways that are still being worked out. The biology here is genuinely unfinished. Not in the sense of being wrong, the core mechanism is well established, but in the sense that the full extent of what peripheral clocks regulate and how tightly or loosely they are coupled to the central pacemaker is still being mapped. I find that honest rather than unsatisfying. Consider the signal chain. One more time. The suprachiasmatic nucleus receives light information from the melanopsin-containing retinal cells. It adjusts its firing pattern. It signals through neural and hormonal pathways, through the pineal gland via the superior cervical ganglion, through the hypothalamic pituitary adrenal axis, through direct autonomic projections to peripheral organs. These signals reach the liver, the gut, the skin, the immune tissues. The molecular clocks in those tissues receive the hormonal messages, melatonin, cortisol, temperature, glucose, and use them as entrainment signals adjusting their phase to stay synchronized with the central clock. By the time the signal reaches a keratinocyte in the skin of your forearm, it has passed through the retina, the optic nerve, the hypothalamus, the pituitary, the adrenal gland, the bloodstream. It has been translated from photons into neural firing patterns, into hormone release, into blood concentration gradients, into receptor binding, into gene expression changes. Each translation introduces a slight delay, a slight imprecision. The keratinocyte's clock is not synchronized to the nanosecond with the suprachiasmatic nucleus. It runs an approximation, a version of the central time filtered through the body's long signaling chains, carried in the concentrations of molecules rather than in direct neural instruction. The approximation is good enough to organize the biology, but it is an approximation. This is the body's time then, not one clock but thousands, not a single synchronized signal but a distributed, imperfect, constantly being corrected approximation of what the master clock in the hypothalamus has decided the time is, which is itself an approximation of the actual light-dark cycle outside, which is itself a consequence of the Earth's rotation. A cascade of approximations, each one close enough, each one serving its local biology, each one running through the night in cells that have no awareness of running anything. The perproteins are accumulating somewhere in your liver right now. The skin cells behind your knee are at a different phase than the cells in your gut wall, which are at a different phase than the cells in your cornea. All of them offset from each other by small margins, all of them running the same loop at slightly different tempos, loosely held together by the hormonal broadcasts of a tiny nucleus that is itself responding to light it received hours ago from 3,000 retinal cells that were simply counting the dark. The loop will close in a few hours. Purr and cry will enter the nucleus. Clock BMAL1 will be inhibited. The proteins will be degraded, the inhibition will lift, and the whole thing will begin again. As it has, in one form or another, in organisms with circadian clocks, for something in the range of 1 billion years. Before eyes, before nervous systems, before anything that could be called a brain. The molecular clock predates all of it. Its earliest evolutionary origins are in single-celled organisms responding to the predictable daily cycle of UV radiation. A clock not for sleep, not for hormone release, not for temperature regulation, but simply for timing DNA replication to occur at night, when UV was absent, when copying errors from radiation damage would be less likely. The clock was a damage avoidance strategy first. Everything else came later. The elaborate orchestration of mammalian physiology, the coupling to the light-dark cycle through the retina and the suprachiasmatic nucleus, the hormonal broadcasts and the peripheral tissue clocks and the temperature rhythms and the immune trafficking, all of this was built on top of a very old, very simple loop that just needed to know when the sun was up. Your cells are still running that loop tonight, in the dark, counting time, waiting for the signal that says the sun has returned. Not yet. You haven't crossed any line yet. That's the thing about this threshold. It doesn't announce itself. There is no moment you can point to later and say, there, that was the exact second. The transition from waking to sleep is not a door. It is more like a shoreline where the water keeps moving and the boundary between wet sand and dry is never quite where you think it is. The neuroscience of this is, in certain ways, stranger than the neuroscience of anything else in this telling. Waking is not the brain's default state. This is one of those facts that tends to land differently on different nights. Waking requires active maintenance, a continuous broadcast of wake-promoting signals from multiple brainstem and hypothalamic nuclei, each one contributing to the arousal state that makes coherent experience possible, the locus coeruleus already described. the dorsal raphe, producing serotonin, the tuberomammillary nucleus, producing histamine, the basal forebrain, producing acetylcholine, and, critically, the lateral hypothalamus, a region containing neurons that produce a neuropeptide called hypocretin, also known as orexin. Orexin is the stabilizer. Its role is not simply to promote waking, but to prevent the sudden, inappropriate transitions between waking and sleep that would otherwise occur. It holds the wake state together. It inhibits the sleep-promoting neurons of the ventrolateral preoptic area, a small nucleus whose activity is both a consequence and a driver of sleep onset, and it maintains the mutual inhibition between wake and sleep systems that keeps each state stable once entered. The flip-flop switch, some researchers call it. The orexin system as the latch that keeps the switch from flipping randomly. Across the evening, orexin neuron activity decreases. The circadian signal plays a role here. The suprachiasmatic nucleus modulates orexin activity among its many downstream effects. Adenosine, which accumulates in the brain across the waking day as a byproduct of neural energy metabolism, also suppresses orexin neurons. The sleep pressure that has been building since you woke, the homeostatic drive that makes sleep increasingly likely the longer wakefulness continues, is partly the story of adenosine accumulation. Caffeine, incidentally, blocks the adenosine receptors rather than reducing adenosine itself. The pressure is still there under the caffeine. The signal just isn't getting through. Without that blockade, the adenosine signal reaches its targets. Orexin activity declines. The latch begins to loosen. The ventrolateral preoptic area, held in check by orexin during waking, begins to increase its activity. Its neurons produce GABA and galanin, inhibitory neurotransmitters that suppress the very wake-promoting regions that were, until recently, keeping it quiet. The inhibition runs in both directions. Wake systems inhibit sleep systems. Sleep systems inhibit wake systems. What changes at sleep onset is the balance. The sleep systems, increasingly disinhibited as orexin declines, begin winning the mutual suppression. The flip-flop flips. Not instantly, not cleanly. The transition zone, Pineal, the hypnagogic state, the period of falling asleep, is characterized by a system in flux, neither clearly awake nor clearly asleep. The two states competing at the level of thalamic circuitry and cortical connectivity. The thalamus is where the sensory relay changes. During waking, thalamic relay neurons fire in a tonic single spike mode, transmitting sensory information faithfully to the cortex, maintaining the continuous sensory feed that waking consciousness depends on. At sleep onset, these neurons shift to burst firing mode, producing rhythmic bursts of activity interspersed with silence, generating the sleep spindles visible on an EEG, and in doing so reducing the fidelity of sensory transmission. The world doesn't go away. The thalamus stops relaying it accurately. This is the mechanism of the feelings, if you have ever noticed it, of sounds becoming less distinct near sleep. The conversation from another room that was comprehensible 20 minutes ago becoming just tonal, just rhythm, just the texture of language without its content. The thalamus is still receiving the signal. It has changed how it's handling it. The default mode network. During waking, this large-scale brain network, involving the medial prefrontal cortex, the posterior cingulate, the angular gyrus, among others, is associated with self-referential thought, mind-wandering, the construction of narrative about oneself and others. It runs when external task demands are low. It produces the stream of internal experience that fills the gaps between focused attention. Near sleep onset, the default mode network begins to decouple. Its regions lose the coherent functional connectivity that characterizes waking. The self-referential stream becomes fragmented, thoughts beginning and not completing, narratives starting and losing their thread. The sense of a continuous observing self becoming intermittent. You felt this. The thought that seemed important a moment ago that is now just the thing you were going to, the hypnagogic state technically, the threshold zone, its contents, the brief visual images, the lurching sensation, the half-formed thoughts that arrive with unearned certainty. These are the products of a system that is neither fully running nor fully stopped. The cortex is still generating activity. The thalamic relay is becoming unreliable. The default mode network is fragmenting. The result is experience without a stable observer to experience it. Something like dreaming, but not quite. Something like thinking, but looser. Something like the people who come back here night after night, who find something in these long, slow biological journeys that makes the dark feel less empty and more inhabited. They're the reason this space has continued. The channel memberships that keep the hours of research and writing possible. The super thanks that arrive sometimes with notes that are genuinely hard to read without feeling something. The quiet act of pressing like, which costs nothing and means more than it should. The subscriptions that say, I want to keep returning to this. They make it possible to keep making it. Thank you. And the orexin neurons are nearly quiet now. The VLPO is running its inhibitory program across the wake-promoting nuclei that kept today assembled. The thalamus has shifted modes. The cortex is receiving a degraded relay and constructing something loose and strange from it. The glymphatic fluid is moving through the interstitial spaces. The molecular clocks in the liver and the skin are accumulating their proteins. The peripheral blood pressure has found its nighttime floor. The hands are warm. The iris is open in the dark, patient, waiting for something that isn't coming, because patience that old doesn't require an object. The perproteins in the cells behind your sternum are still counting. The astrocytes in the hippocampus are still, the thoughts are arriving without. There was something about the thalamus, the relay becoming the sense of an observing self that keeps. Something in the brainstem still sending a signal, quieter now, and the cortex receiving less of it, constructing less from it. The narrative becoming, the proteins accumulating in the warmth still in the hands, the iris still, the dark that the skin's opsin proteins registered first, hours ago, before any of this had a name, before the eyes, before the signal moving through the long chain of